Skeletal muscle fibers contract as a result of electrical impulses from nerve cells called motor neurons. To understand how muscle contraction works, we divide the entire process into 3 phases. The first phase is muscle excitation at the neuromuscular junction. This is covered in the IP 2.0 topic, Muscle Excitation at the Neuromuscular Junction. The second phase is excitation-contraction coupling, the focus of this topic. And the third phase is cross-bridge cycling, which is covered in the IP 2.0 topic, cross-bridge cycling. Remember that muscle excitation at the neuromuscular junction ends by initiating an action potential on the sarcolemma of the muscle fiber. Excitation-contraction coupling is the sequence of events by which the muscle fiber action potential, the excitation, leads to muscle fiber contraction. First, we will examine the structures of a muscle fiber that are responsible for excitation-contraction coupling. These structures include the sarcolemma, the transverse tubules, the sarcoplasmic reticulum with terminal cisterns, and the sarcomeres of each myofibril. Let's begin with the sarcolemma, the cell membrane surrounding the muscle fiber. As we saw in muscle excitation at the neuromuscular junction, a somatic motor neuron stimulates a muscle fiber at the junctional folds, initiating an action potential on the adjacent sarcolemma. Voltage-gated sodium channels in the sarcolemma conduct the action potential over the whole muscle fiber. The transverse tubules, or T-tubules, are regularly spaced tube-like infoldings of the sarcolemma. They branch within the muscle fiber to reach each myofibril. Since they are infoldings of the sarcolemma, the T-tubules also contain voltage-gated sodium channels. When these channels open, they help conduct the action potential from the muscle fiber surface down into the muscle fiber interior. Adjacent to the T-tubules are sac-like bulges called terminal cisterns of the sarcoplasmic reticulum. The sarcoplasmic reticulum, or SR, extends outwards from the T-tubules, forming a web-like network surrounding each myofibril. The membranes of the T-tubule contain voltage-gated proteins. These proteins link to calcium release channels in the terminal cisterns. Together, the voltage-gated proteins and the calcium release channels control the release of calcium into the sarcoplasm. The calcium ions from the sarcoplasmic reticulum activate the sarcomeres of each myofibril. A sarcomere consists of alternating thick and thin filaments. The thick filaments are made of the contractile protein myosin. The thin filaments consist primarily of the contractile protein actin. The actin of the thin filament has binding sites to which myosin attaches during muscle contraction. 2 regulatory proteins, tropomyosin and troponin, are also associated with the thin filaments. Tropomyosin blocks the binding sites on actin when the muscle is not stimulated. Troponin holds tropomyosin in place over the binding sites. Calcium binding to troponin causes tropomyosin to shift away from the binding sites. Now that we know the components responsible for excitation-contraction coupling, let's learn how a muscle action potential causes the release of calcium ions from terminal cisterns of the sarcoplasmic reticulum. We'll start with the action potential initiated on the sarcolemma. As you learned from the IP 2.0 topic, muscle excitation at the neuromuscular junction, there is a resting membrane potential across the sarcolemma that results from the distribution of ions across the membrane. In particular, sodium ions are more abundant outside the muscle fiber than inside it. Activity at the NMJ triggers an action potential in the adjacent sarcolemma by opening voltage-gated sodium channels. As sodium ions enter, depolarization spreads to affect nearby voltage-gated sodium channels on the sarcolemma. These channels are called voltage-gated because they open when the voltage across the membrane reaches a specific threshold value. When they open, sodium ions diffuse inward, depolarizing the adjacent membrane and triggering an action potential in this new segment. This depolarization spreads to trigger additional voltage-gated sodium channels in nearby segments, and the action potential spreads along the sarcolemma. Voltage-gated sodium channels in the T-tubules allow them to conduct the action potential throughout the muscle fiber interior, ensuring that all the myofibrils contract simultaneously. Now let's see how the action potential on the T-tubules stimulates calcium release from the sarcoplasmic reticulum. In addition to voltage-gated sodium channels, the T-tubule membrane has voltage-gated proteins that interact with calcium release channels in the membrane of the terminal cisterns. These voltage-gated proteins respond to depolarization by changing shape, opening the calcium release channels. Calcium ions rapidly diffuse out of the terminal cisterns through the open channels and flood the sarcoplasm of the muscle fiber. The key to excitation-contraction coupling is an increase in calcium ions within the sarcoplasm. We just learned what causes the release of calcium ions from the terminal cisterns of the SR. Now let's see how the calcium ions allow the sarcomeres to contract. To understand the role calcium ions play in ECC, we will need to look more closely at the thin filaments. Each actin has a binding site for the myosin heads, but in the absence of calcium ions, these sites are covered by tropomyosin. Calcium ions released from the terminal cisterns bind to troponin. This causes troponin to change shape in a way that shifts tropomyosin away from the myosin binding sites on actin. Once the binding sites are exposed, the myosin heads can bind to them, forming cross bridges between the thick and thin filaments. Muscle contraction can now occur through cross-bridge cycling. This process is covered in the IP 2.0 topic, cross-bridge cycling. Let's review what we have learned. Stimulation at the neuromuscular junction triggers action potentials that spread across the sarcolemma and down the T-tubules. In the T-tubules, the action potentials cause voltage-gated proteins to undergo a shape change that opens calcium release channels of the terminal cisterns. Calcium ions released from the terminal cisterns bind to troponin, causing a shape change that pulls tropomyosin away from the myosin binding sites on actin. Myosin heads can now bind to actin, and the muscle fiber contraction can occur. As a result, calcium ions are responsible for coupling electrical excitation to the contraction of skeletal muscle fibers.